Hello and welcome. My name is Vanita Dahia, and today we're talking to you about conquering chronic illnesses, particularly in relation to toxic exposure and the rather topical topics of Lyme, mold, and various biotoxin exposures. So what you learn in the session is about a little bit about the introduction to molds, biotoxins, Lyme infections, some of the environmental toxicants, particularly the endocrine system and how that gets affected and how they get disrupted, and a little bit about the gut and the microbiome. Unfortunately, due to time constraints, we're going to focus on the various testing opportunities available, particularly in chronic illnesses. Now, chronic illnesses is not a linear disease. It's not an iron deficiency, so you give an iron tablet or an iron infusion. Chronic illnesses may incorporate autoimmune diseases, and they do impact multiple organ systems. So we need to, as we walk through the clinical door, need to look at and examine all the various clusters of symptoms affecting all the various organ systems. They may affect the adrenals, genetics. It is important to identify the various gene aberrations, the effect on the endocrine system. So we're referring to hormones, the thyroid, the adrenals, and obviously the immune system lives in the gut. So if you have a chronic illness, the immune system is impaired, the gut always needs to be addressed first. The effect of chronic illnesses affects mental health, so we need to obviously manage that effectively as well. The most of the webinar today is really about molds, pollutants, pesticides, and various infections. We definitely talk about methylation and environmental pollutants because they also affect the immune system. Sometimes you may not be bitten by a tick and you may not have Lyme disease, but there may be other sensitivities that can contribute towards your chronic illness. The adrenals play such a vital role in chronic fatigue syndrome, in fibromyalgia, etc. So when you're identifying somebody who has a chronic complex condition, invariably they would be stressed. Invariably they would be fatigued. So signs and symptoms of adrenal dysfunction can vary from total fatigue or fluctuations of energy and the capacity to be able to crave certain foods such as sugars or salts, but then there are other symptoms associated with adrenal dysfunction as well. Cortisol is the main stress hormone, and when we identify cortisol, it is done through saliva, through serum, through urine, through blood spots. So there's a number of opportunities to measure the adrenal function. The idea about measuring adrenal function is to establish the stage of adrenal fatigue or adrenal insufficiency or exhaustion. So there's three major stages, and when you're exhausted, both cortisol and DHEA become flatline low, and that is part and parcel of adrenal fatigue. So that's total exhaustion. So cortisol affects all organ systems. It leaches the calcium out of the bones. It sends signals to the brain to stimulate adrenaline rush. And most importantly, cortisol is a stress hormone which will allow the body to react to the stress immediately. So at the expense of all other organ systems. So when the body is under stress, the body requires energy to react to that stress. The energy is easily obtained from glucose, and glucose is easily obtained from muscle tissue. And this is why some patients will tend to have muscle fatigue, aches and pains when they're under stress or they have a chronic illness, because the liver stimulates gluconeogenesis to provide that level of energy to combat that stress. This is a typical result of a saliva cortisol test. It's usually measured at different times of the day, 4 times in a day, and in addition, it can be measured in the morning as well. And so the idea behind measuring cortisol at different times of the day is to establish, did you have enough sleep at night? Are you wired and tired, or are you feeling refreshed in the morning? Another component is, do you have reactive hypoglycemia, perhaps later in the afternoon? And as you can see, this person's cortisol is high in the morning and high at night. So this person's firing on all cylinders in the morning and at night. So obviously, I would question the sleep patterns. Methylation plays a really good or a significant role in every metabolic process. Now, if you understand the methylation profile, then we can probably shut up shop and stop the talk right now. The methylation is really the basis for life. Methyl groups are a carbon and 3 hydrogens, and they are either donated through various pathways. The 2 major pathways that are significant are your folate pathway, that's the top circle, as well as the SAMe pathway. And all the markers that are in green are your enzymes. These are MTHFR, methylene tetrahydrofolate reductase, MTR, MTRR, which is methionine synthase, and so forth and so on. So every pathway requires specific nutrients for methylation to occur. Methylation is necessary to produce phospholipids, to produce energy, to produce hormones, to produce neurotransmitters. And so the idea is to rectify and regulate homocysteine. Too high, problems with cardiovascular disease, cancers, inflammatory conditions. Too low, inability to produce energy in the form of glutathione. So typically, most chronic patients might be familiar with MTHFR. That does not answer all the questions in relation to methylation. Ideally, you would want to measure all your genetic markers associated with methylation. Methionine synthase is dependent on vitamin B12, magnesium, B2, etc. Methionine 5-MTH reductase. It's a homocysteine reductase. In other words, homocysteine to the folate pathway. Again, it's dependent on B12 and really good amounts of betaine hydrochloride and so forth. COMT is a catechol methyltransferase, not only involved in methylation in the methylation pathway, but it is also necessary for metabolism of hormones as well as neurotransmitters. So we are born with these genes, but we can change. So we can't change the actual gene, but we can change its expression. So a typical result might look like this, where you will see the markers that are considered to be present in terms of mutations. So MTHFR, one or more mutations might be present there. Examine what they are to give us an indication as to the degree of methylation defect, which nutrients would support MTHFR, and that would be a methylated form of folic acid. There are one or more mutations are found in COMT, MTRR, and MTR. So definitely we need to put those nutrients back into the system. So it is important to actually look at and identify all the markers associated with methylation profiling. I find that when I deal with chronic inflammatory conditions, I put in the mop-up phase first. In other words, correct the adrenals and support the basic nutrients, for example, amino acids, vitamins, and minerals right up front before we start addressing all the infections, because patients who are chronically fatigued cannot tolerate all the various medications that are needed to address Lyme and mold, etc., without a a decent amount reserve of energy to be able to cope with it. That energy is obtained from amino acids, various minerals, and a good adrenal function. So amino acids are actually precursors. They are breakdown products of protein, and they are precursors to neurotransmitters. They are necessary for liver function. They're necessary for muscle function. They're necessary for every physiological function that our body needs. Mitochondrial function requires heaps of amino acids. Histidine is an amino acid that eventually makes histamine, which is a neurotransmitter. So there's all these neurotransmitters are derived from amino acids. Typically, amino acids can be measured either in urine or it can be measured in a plasma sample. Ideally, you would require a compounding pharmacist to make up an amino acid formulation for you, somebody who's trained in this area. And please contact me if you are interested in this area, because we can help you in this area. Firstly, amino acids need to be optimized. If they are not optimized, you will see huge physiological imbalances in every single pathway. Glutamine is it's the most abundant amino acid found in the gut. It is necessary for the gut integrity, but it is also needed for GABA production. So customization of amino acid is very, very important. And I'm not talking about these large containers of commercially available amino acids, which are adulterated with lots and lots of flavors and colorants and additives and bulking agents, etc. We're talking about amino acids, pure raw material that is individually compounded based on your results. So I'm happy to discuss that further. Now, it's very important that patients who have chronic and complex health conditions may have been exposed or terribly sensitized to environmental pollutants and toxicants. It could be some of your fluorocarbons found in your ventilation ducts, or your tricresols that are part and parcel of aerotoxic syndrome. We're flying so much these days, and that's affecting our the air that we breathe. Industrial cities with huge amounts of smog. People are inhaling that all the time. So chronic complex health conditions, people become really sensitive. Often our carpets and furniture all contain various chemicals. So how do we identify these chemicals? Conditions can be associated with certain chemicals. For example, chronic fatigue syndrome may be associated with various forms of toxicities, ranging from arsenic, benzene, cadmium, formaldehyde, lead, and so forth. And so there is a correlation in terms of certain diseases. Alzheimer's and aluminium is fairly well known. Glyphosates, which we know as weed killer, is probably found in most of our produce today. Even our animals are eating produce that has been laced with glyphosate. So glyphosate is now well known to be not only damaging to the nervous system, but a possible carcinogen. So pollutants can actually be measured in a urine and a blood sample, and there are numerous tests. For example, if you have you're a farmer and you're having a lot of chronic health conditions, you might want to examine the level of chlorinated pesticide exposure. If you're a hairdresser or a mechanic, you might want to examine the volatile solvents. So each of these panels are relatively important, and I will bring your attention to just one panel to give you a peek at part of the testing. Alkyl phenols are are found in various beverages, even your till slips, your thermal sail slips, water bottles, plasticware, shaving creams, cleaning supplies. They all have some of these alkyl phenols, and they are found everywhere. They even are effective as an oil and water-based surfactant. They're found in detergents, and they even in our homes. So bisphenol A, for example, non-alphenols in this particular test result, your non-alphenols are particularly found in those thermal till slips, and that's on the higher side. So it is very, very important to actually remove that exposure, remove the exposure of perhaps the cling wraps which contain non-alphenol, microwave plasticware that contains various BPA products. And BPAs are now associated with hormonally estrogen receptor positive cancers in both men and in women. Now, molds and biotoxins, mycotoxins, gliotoxins, Lyme and its co-infections are rather topical because we are exposed to what we call the sick building syndrome. We're exposed to various molds. People are traveling, they're traveling to areas that they've never been to, and they might have been bitten. It could be a Western society, it could be a remote Amazon type area. But the key component is most people can handle all of these infections. They can handle the mold. They will find that some patients, you will find that some family members will not be affected since moving into this new house that may have been infected by mold. But if your immune system is rather poor and you already have an autoimmune condition, anything like Hashimoto's, for example, then that person's immune system is already compromised. That person is likely to react to the smallest amount of toxic exposure in any form, whether it is mold or whether it is a bite. So they stimulate these various inflammagens and lead to a cascade of chronic inflammatory responses. And CIRS is a term that is now coined as an acute and chronic systemic inflammatory response. And this syndrome is associated with water building damage, with toxigenic organisms, with various microbes, be it fungi, bacteria, or viruses. So this is quite a common occurrence these days, particularly in chronic conditions. Many bugs are involved. Fungi, which actually form your spores, they grow indoors and outdoors. They thrive in very damp environments, and there are many types. Cladrosporum, penicillin, and aspergillus are very, very common. There is a test that can actually measure, it's a blood test that measures for various types of mold. And so you can identify that mold, or you can actually identify it through another mechanism. So the idea is to identify all the various components that are associated with molds, beta-glucans or polysaccharides of your glucose monomers. Inflammagen elicits your pain, your inflammation, your edema, and the cellular response to inflammagen. So you'll hear some of these terms as you start to investigate in this area. Microbial volatile organic compounds, known as VOCs. These microbes release organic compounds and they give a very distinctive musty odor. And then what they do is they activate your innate immune responses in these susceptible. So your innate immune response then is affecting the adaptive immune response. So you will find that patients that originally have a bacterial or a fungal infection might have had a significant viral load to start off with. Often the term chronic fatigue syndrome was first originally classified as a chronic viral infection, chronic EBVs, chronic CMV. There are many, many bugs that are contributing to where we are at the moment in chronic complex conditions. So water damage to buildings, WDB, is associated with obviously growth of fungi, bacteria, mycobacteria, actinomyces, and you'll find that not only where it's visible, but also where it's invisible. So it could be, complex mixtures of various contaminants that are present, and they will present itself in various forms of symptoms. The symptoms that you typically would see in a chronic complex condition is usually fatigue. That's the first thing. And then as a result of the frustration of not being able to do anything, it affects the system neurologically. People will have allergies, whether it be a red eye, sinusitis, rhinitis. The aches and pains are invariable. So they might have joint pain, they might have skin sensitivity, mood swings, appetite changes, shortness of breath, abdominal pains, etc. So the symptoms are vast, and it often is misdiagnosed with various other conditions. So conditions such as chronic fatigue syndrome, MS, depression, stress, allergy, or PTSD. So if you have a chronic complex condition, where would you start in terms of identifying what are the opportunities, and where do you get your best bang for your buck? Because there are so many tests out there. I'll bring your attention to something that I think is very, very important, because there are far too many tests out there, and you must ensure that the labs that are used are accredited in terms of their international lab standards, because they can vary from lab to lab. The first test that I think is worth mentioning is your human leukocyte antigens. We'll talk about that in a bit more detail, but that gives us the real umbrella view of what's happening. Is this patient exposed to an infection? Is there Lyme disease? Is there post-Lyme? In other words, did they have Lyme? Were they bitten in the past? Do they have mold, etc.? If the patient has a nasal congestion, sinusitis, rhinitis, and they may not necessarily have any of those symptoms, but they might be multiple antibiotic resistant, have coag-negative staph infections. So you can measure just a little nasal swab, pop a paddle in the nose and extract a sample of the nasal mucosa so that the lab can test both bacterial and fungal infections. If a patient has moved, for example, has moved in a workplace, in a home, and they find that there's mold there ever since they've moved there, they felt sick, and suddenly discover, look, there's definitely something going on in the structure, they can actually remove a sample of that particular mold or dust or cement slab or whatever building equipment that they can in order to have that tested. In addition to that, you can actually look at specific mycotoxins and identify them as well. In the case of Lyme, it is very important to look at the PCR as well as the West. The more technologies that you use to identify not only Borrelia and all its species, but your Babesia family and all its co-infections. That's the test that you would probably deal with. It's a tick-borne disease immunoserology together with a Western blot. That's a good starting point. Later down the line, you can do individual testing as a retest. So the HLA studies are basically, HLA stands for human leukocyte antigen, and these antigens live on every cell. They provide the immune responses for your body. Now, typically, we had HLA DQ B27 has been typically associated with conditions of certain conditions such as ankylosing spondylitis. Typically, HLA-DQ studies have been associated with identification of celiac disease or gluten intolerance. But now the works of Dr. Horowitz, Dr. Richie Schumacher, Dr. Stephen Brunner, these people are founders in amazing research. And Dr. Schumacher, in his book called Surviving Mold, has actually developed what we call a Rosetta Stone. So what he does, he takes the DQ studies and the DR studies and matches up the various bands and the expression of the various haplotypes to be able to identify a specific outcome. So the typical test result might look like this, where the HLA-DR studies, in this case it's 11 and 15, DQ, A1 and B1 are the various alleles, 6 and 7, 1 and 5. So you're matching up the DR and the DQ. And as you can see, 15 in the DR and 6 in the DQ is expressing a haplotype in DRB5 as the haplotype number 51. So 15, 6, 51 is associated with Borrelia or post-Lyme syndrome. So this patient has a Borrelia infection. Which one? We don't know. So we need to do the Western blot to determine which one. Post-Lyme syndrome, this patient has been exposed to a tick and they have been exposed to Lyme in the past, and they're exhibiting all the signs and symptoms right now. And in addition, multisusceptible antibiotic resistant stack, this patient has MARCONs. So now it gives us an opportunity. Should we do the nasal swab in this person? Yes, we do. So we can identify which bacteria and which fungi this patient might have. And so you can tailor your specific antibiotic regime accordingly. The biotoxin journey is a great resource. There's plenty of resources out there, so welcome to go and have a look at it. Now, the susceptible haplotypes that are expressed in, let's say, in a chronic Lyme will be 5,651 or 16,551. So these are expressed and well documented. The biotoxins, they're usually tagged, and the mold patients don't have an immune response or the specific HLA genes that are required to form this antibody to fight against that foreign antigen. So then the biotoxins then start to bind to these receptors, leading to a bunch of inflammation. That inflammation is measured through various Lyme studies, and we're identifying your various complements, your TGF-beta1, MMP9, matrix metallopeptase. So when you see a high C4A, if C4A is a certain number, if it's under 3,000, it's between 2.5 and 3,000. Some texts have shown that it is associated with Lyme. If it's above 3,000, then it may be associated with mold. The jury's still out. The whole area of identification and quantifying all the various susceptibilities is still very much, even though it's rather in-depth, but it's the study of Lyme is in its infancy. So as more material comes about, we'll be informing you as we go along. In addition, you could look at VIP, MSH, and vasopressin. These are markers. It requires much further time to discuss each of these markers, but it is expressed perhaps in Dr. Schumacher's biotoxin pathway. Now, if you have an opportunity if you are exposed to Lyme, it's a good idea to have a clear understanding of how all these various insults affect the hypothalamic markers of VIP, MSH, and AVP. And it'll all affect various areas, whether it is gastrointestinal, neurological, etc. So the sorts of testing, for example, MSH, each of these markers have its value. It's important to understand what each marker does, but 95% of mold patients tend to have low MSH. ERMI scores are a different test, and it really stands for Environmental Relative Moldiness Index. So it's telling us the level of mold in our body. So the ratio of water damage to common indoor molds is typically between 10 and 20. So if you have an ERMI score that is, if it's 2 or less than 2, then the patient is likely to have CIRS. There are other markers like C4A greater than 20,000 may be associated with mold as well. So this is a typical ERMI result where it is actually measuring the specific mold toxins, the biotoxins, and it'll express as an ERMI score. EMMA, on the other hand, is environmental mold and mycotoxin assessment, is a more updated version of ERMI, and the combination of those will be valuable for us to establish level of water saturation. Do we need a mold? Is this patient suffering with mold? Which are the bugs that are causing the mold? And what do we do about it? Now, doing something about molds and mycotoxins is very, very important because you have to. The first thing, if you've established that you do have mold, you have to move away from the source of that exposure. Might have to move house, might have to clear out all your things that have been exposed to the mold. So mycotoxicosis is really the ingestion of the contaminated foods that have been exposed to the mold. So don't think it's only in the environment. You can consume these mold toxins as well. So your mycotoxin producing fungi and mold, they are tested by real-time labs, and there's a number of these various species and mycotoxins. Typical result might be expressing in the form of parts per billion, and they may either be negative or positive. Lyme disease, on the other hand, is very typically chronic. It's chronic fatigue, it's lethargy, may be associated. Not all chronic fatigue patients have Lyme, and not all Lyme patients have chronic fatigue. It's very important to identify the clusters of symptoms, the underlying cause. So assessment, we cannot guess this. No practitioner is able to guess this. We have to test whether you're either positive or you're negative for Lyme. That's it. You're either positive or negative for mold. So a Lyme patient likely to have inhibited cellular function and protection. The innate immune system is impaired. You'll find that the systemic bacterial infections will be repeated. The patient's likely to have a Borrelia burgdorferi bacteria that's transmitted. Typically, you'll see the erythema migrans, which is like your butterfly skin rash, and a lot of lethargy and flu-like symptoms. A typical result might look like this, where you will see your burgdorferi, IgG, IgM, and their various co-infections in units. The Borrelia subspecies are also identified, as well as the various Babesia, Ehrlichia, Bartonella co-infections. So the patient might not have Lyme, but they might have the co-infections. And the result is usually expressed in various forms of bands, and if 5 or more bands of one are present, then that person may be positive or reactive. So in a nutshell, a differential diagnosis is most important. You cannot guess this. You can identify through a number of questionnaires, but testing is absolutely essential. The second thing that we need to be aware of is the correcting toxic carriage, correcting elevated MMP9s, the replacing low VIP, etc. So there's a whole bunch of strategies that need to be addressed. So what is the prevention and what is the intervention? One of the major areas in terms of identifying mold is to do what we call a visual test, and that's called a VCS aptitude test. It's an online screening tool that measures one of the neurologic functions of the visual called contrast, and it assesses and documents capillary hypoperfusion from these neurotoxins. So that's a really great, easy, cheap tool to actually use at the offset. In the CIRS case criteria, we need to actually identify the biotoxin illness. You use a symptom questionnaire. There's a number of questionnaires, but based on Dr. Schumacher's work, there are eight symptom clusters is consistent with biotoxin illness. And if you see an abnormal rise in the arterial hypertension during exercise, and then you'll see this neuroregulatory, neuroendocrine, neuroimmune dysfunction, is inflammation, there's endocrine abnormalities, you're likely to see a typical CIRS case. The patient, you definitely, as a practitioner, need to understand the patient's history. What is the patient's exposure to the wet building syndrome? Has the patient traveled? Have they gone off into a river and swallowed a whole bunch of water in an infected river? So the haplotypes, the HLA test is probably a good starting point before you go out and dig deeper into the specific tests based on the results of HLA. You make the determination. If MARCONS is not present, no point measuring MARCONS. So these are a strategy, Dr. Schumacher's work on surviving mold. There's various strategies on day 1, day 2, day 3, etc. And you will see various treatment protocols that will affect your C4A markers, VEGF, and so forth. So the treatment steps are predominantly removal from exposure. Secondly, start using various mopping up agents like chelating agents, cholestyramine, Welchol, bentonite, charcoal. These are strategies that will start to mop up in the gut, and so that you can actually restore some of the inflammatory or stabilize the inflammatory markers. And then you start addressing every piece of the pathway. Eradicate the MARCONS. It might need VIP or BEG spray, or you could look at some of the natural formulations, which may contain colloidal silver, turmeric, garlic, and various essential oils. The prevention remedies it's not a one-step process. When we're talking about inflammation, we definitely need protection, that's for sure. We need removal from exposure. We need lots of education in this area so that patients can start to identify prevention, they educate, and then they start to intervene. The intervention strategies can either be pharmaceutical or they can be naturopathic, and ideally you combine the two. The idea is to reduce your airborne exposures and to reduce the foodborne exposures as well, because they are contained in our foods and in our agricultural crops. They contain various moisture. Crops contain moisture during both growth and post-harvest, so you need to be very careful what you eat as well as what you're exposed to externally. So the step-by-step guide is really identifying, head-to-toe exam, look at the signs, symptoms, do the questionnaires, check the initial labs, the HLAs, and then start looking at the specific markers, and then also removal from exposure. The exposure can be through Borrelia, from dinoflagellates, food poisoning, or from exposure to the interior or water-damaged buildings. So there's a number of opportunities. The side effects of a lot of these drugs you need to be aware of, and besides that, patient will tend to have their own level of symptoms as well. So it's important as you're treating a patient, make sure that you're examining how a patient is responding with every step of the pathway. You can't throw a chelator as well as a whole bunch of antioxidants. It's always a step-by-step approach. So the eradication of the MARCoNS is very, very important. These bacteria form the biofilm. So if there's a biofilm in the gut, for example, you might need to use specific kinases like nattokinase, lumbrokinase, teprokinase to break down that biofilm in order for the antibiotics to work or any antimicrobials to work. Colloidal silver has now been shown in a study together with garlic to be very beneficial as an antimicrobial in the nasal mucosa. Various antibiotic regimes are also used, rifampin, amoxicillin, and there's a whole, your fluorocarbons, etc., all used, your fluoroquinolones, sorry, are also used. The very important thing is that there are huge amounts of study on the value of certain antioxidants such as coenzyme Q10, B group vitamins, cordyceps, turmeric, anti-inflammatories. These are really very, very important, not only to stimulate your immunity, but also manage the inflammation. We need to improve the energy production, which is your ATP production. We need to be involved in immune balancing and boosting the resistance to the infection. We need to ensure that probiotics are used because they will disrupt the pathogenic bacteria and protect your commensal groups. It'll encourage the restoration in the event of any disruption, and it'll enhance gastrointestinal mucosal integrity. That's so important. We need the right amount of immune function in the gut, and so we can have the beneficial effects of not only probiotics but omega oils. The essential fatty acids are actually protective to the body. So there's a whole range of tools that can be used in both the natural as well as the pharmaceutical routes that can play a major role in treatment. Various oils such as frankincense, bergamot, oregano, thyme have also been shown to be very beneficial. Foods that are rich, like bone broths, rich in various amino acids, have also been found to be useful. Commercial items such as a product that I use routinely called Lymex. It's based on traditional Chinese medicine. Lots of study been shown to be very beneficial as not only an antispirochetal, but it's antibacterial, antifungal, antiviral. So great to have a multimodality formulation that's natural, that does not affect the system or have the side effects most of your pharmaceuticals might have. The chronic illness natural interventions, look, it's not a one-step process. Seriously, if you've got a chronic condition, it is so important that you need to consult with an expert in this area. All I've done is given you a very brief overview of all the various insults that can happen from a toxic exposure or from an infection perspective, what you can do to test, what a test result might look like, but this is really only the tip of the iceberg. And all the professionals out there that are working in this area, even though they may be experts, we are all taking baby steps because this area is a burgeoning area. It's affecting so many patients. We are constantly learning. So definitely consult with a qualified practitioner or somebody who is familiar with this area. This is your health. Take action, and I am available for consultation, so you're welcome to give me a call. Available at this various pathology lab, a clinic, or welcome to glean some of this information out of the book that I've written on mental health. So I thank you so much for your attention, and I look forward to touching base with you. Welcome to ask questions, and catch you next time. Thank you.